Welcome to my PS121 media project. I'm going to be talking about a study done by Ting Sui and colleagues on novel rabbit model of Duchenne muscular dystrophy generated by CRISPR Cas9. DMD stands for Duchenne muscular dystrophy. It's a disease that affects the sarcolemma membrane in the skeletal muscle. The dystrophin protein connects the extracellular matrix and the cytoskeleton, which helps in stabilizing the sarcolemma membrane and protects skeletal muscles from contraction-induced injuries. Without the dystrophin gene, there is no binding, resulting in the loss of other complexes, ultimately leading to the leak of creatine kinase to extracellular membrane. There were different animal models used to study DMD. Commonly used is the mice. It's used for comparing wild-type and dystrophin knockout mice. However, mice show a milder phenotype of DMD. Other animal models used were dogs and pigs. The most common dogs used are golden retrievers. Pigs do not show cardiomyopathy, which is unlike the human models. However, both show severe phenotype of Duchenne muscular dystrophy, so it makes it ideal for human clinical trials. One obstacle that they both share is that studying these animal models are very expensive. In this paper, Ting Sui and colleagues focused on rabbit animal models. According to the paper, rabbits are ideal to be used as animal models because they share similarities with humans in terms of genes, anatomy. They also share a severe phenotype of DMD displayed in humans. One advantage of rabbit models is also that they are inexpensive. The team designed a CRISPR-Cas9 that targeted exon 51 because most mutations in DMD patients are in exon 51. This CRISPR-Cas9 sequence was then microinjected into the rabbit zygotes. Then it was cultured into the blastocyst stage. After 128 was microinjected zygotes were transferred to the oviducts of 4 surrogate rabbits. 26 out of 33 pups carried the DMD mutation. Then, the rabbits were tested for signs of Duchenne muscular dystrophy through tests of immunofluorescence for the dystrophin protein, and they also wanted to test if there is occurrence of cardiomyopathy, the same as human patients in rabbits. For the immunofluorescence test, they compared a wild-type rabbit to a DMD rabbit. They tested for the presence of dystrophin as well as alpha-dystroglycan and alpha-sarcoglycan in the muscle. As you can see, the wild-type rabbit showed fluorescence for dystrophin gene, alpha-dystroglycan, and alpha-sarcoglycan. The DMD rabbit, however, did not show fluorescence for these proteins. These indicate that exon 51 mutation caused for a non-production of the dystrophin gene, meaning there's DMD for rabbits. For testing cardiomyopathy, the scientists did an H&E staining for cardiac muscles. They compared biopsies of wild-type and DMD rabbits during their 5 months period and 12 months period. This shows the progression of cardiomyopathy. In wild-type rabbits, the staining showed healthy normal muscle fibers with little variation in size and minimal fibrosis. In DMD rabbits, it showed hypertrophy and fibrosis. It was also noted that fibrosis progressed in a 12-month period compared to the 5 month period of DMD rabbits. Ting Ting and colleagues also measured the ejection fraction of the DMD rabbits through an echocardiography, or EKG. Comparing it with wild-type rabbits, DMD rabbits showed a significantly lower percentage of ejection fraction, which indicates cardiomyopathy. In conclusion, rabbits as animal models for DMD is promising due to the presence of muscle weakness and wasting, showing signs of respiratory disease, showing signs of cardiomyopathy, and dystrophin mutation. They are also inexpensive and they have short gestation period, which is ideal for several testings. All in all, rabbit models are good models that can be used for clinical trials of medicines to treat DMD patients. This has been a media project done by Marianne Tan on the research article by Ting Ting Sui and colleagues published by the Company of Biologists. Thank you for listening.